size.
What a beautiful
I just
fish.
love seeing things like appear out of the darkness. It's just, it's so cool.
I love the diversity of the fishes, especially down here. Like, so many different tail shapes, so there's so many different niches that they hold, and that's really cool. Crazy.
So I don't know if I'm going to be able to go any deeper on that wall.
That's fine.
Because I got the crevice there.
Yeah, yeah. So I think we can stop that Transect survey. And so now one of the things that we want to start doing is sample some of these corals. So I don't know for you what would be. There's several corals, so we would like some of these large bamboo corals that we've been seeing. Some of these as well.
Wow. Just
far,
wow.
I know, I wonder how far it goes.
Look at that.
The upside down, look at that. That's amazing. Jeez.
There's a sea star there. Just so much life.
Let me know if you see anything you want to sample, and then we'll see what we can do.
Yeah. I mean, there's a different that I want to sample here, but I don't know if on the fly is fine for you, or if we should look for a place where you can land. Let's check the top of that structure here, see if there's anything easier. So I think we should try to get some of the things that are the most common. So I've seen so far some of these maybe large bamboo coral, like this one. There's this other coral that we haven't seen yet called Gorgia. There were a few of them on that wall, and I saw a lot at that site, so I think it would be nice to collect that as well. We've collected crysogorgia before, but there's a lot of them here, so maybe that could be worth also getting a colony.
Look at that hole.
If we can get some associates at the same time, like these bamboo corals seem to have a bunch of other things on it, so I think then we can
I know that there's a hole in the top here. And it's full of coral.
Oh, wow. Oh, there's some of the other coral that I'm interested in that hole, actually.
Wow.
Wow, that's insane. Wow.
It's really cool.
I love this site.
Yeah, this site is so amazing.
Yeah, I'm also interested in those ones, but there may be easier targets. I've seen a bunch of them on the wall as well.
Yeah.
What is in there?
I'm just looking in the hole.
Yeah, no. That brown thing.
Can we zoom in actually in that hole?
Let's zoom in the hole.
Okay, zooming.
I can take like a 5-meter step towards Herc.
Oh, we got a fish in the background as well.
There's another fish.
Yeah. Oh yeah, that's an eel.
Oh,
type fish.
Love it.
Cutthroat.
It's
That
kind
looks
of
like a
blurry
pretty big
from here,
one.
so I can't tell much. Look at that. It's a cave.
Yeah.
Are those anemones?
No, wait.
The red one?
Yeah, it could
What
be
in
some
the
kind
world?
of zoanthids growing on a piece of coral, maybe?
That's crazy.
There's like that coral is really hiding.
Wow, it's just like covered
How are
in
they
light.
able to live? They're not able to access marine snow.
Well, clearly they somehow.
Somehow.
Somehow. Yeah, you can zoom out. So yeah, I would like to go for that large bamboo coral that is right ahead of us and collect some of its associate with it. So let's have a look at it and see what we can get.
I'm in love with this site. So much fun. I know, I was about to try to. Okay.
Does that mean anything in Marshallese?
I only know the logoto part. I'm still trying to figure that out. But logoto is basically like it has to do with like something being windy.
Oh.
Because koto means wind.
Okay.
Huh.
That makes sense.
Still trying to figure that out. I'm gonna have to ask around as soon as we get done with this.
Are we sampling this?
So let's zoom in on that coral first. Yes.
Zoom?
We are?
But I think I would like to get as many associates as we can from this.
Is that a crinoid back there?
There's a couple of crinoids on it.
What's that little red
Mike, you should
dot
start feeling
up
the
in the
step
top
pretty
left?
soon.
Do it?
I think it's
You should start feeling the step
Oh.
relatively soon.
There are some anemones on it as well.
Hayden, I'm going to pause the samplers because we've been in this area for a while and I want to, okay, so pausing 0 and 1. No, I'm just pausing these just since we're going to be at this area for a while and we've been sampling for quite a 42 minutes already. So,
This is incredible.
so right now there are no, I'll start it after we're done with all the collections. Yeah.
Sure. All right, let's zoom out and look to the left side of that coral, please.
Zoom out.
Going wide.
Okay, so I was thinking we can get a branch with some of these anemones on it. So somewhere to the left, maybe like one of those lower branches.
On the left side?
On somewhere around here. I need to check where are the lasers?
Okay, go zoom in.
Zooming.
Let's see. Or maybe like somewhere here in the top of the coral.
Okay.
Sorry, did I go too close?
Zoom wide.
Yep.
But that seems like a pretty big piece actually. Let me see if we can get smaller.
This is a bamboo coral, correct?
Yeah. Or maybe because, how about, let's see, can we slurp a crinoid first as an associate and then sample a branch?
Yeah. So you want the crinoid?
Yeah, but I think we'll need to slurp it, otherwise it might go away if we grab the branch.
Yeah. Okay.
So both of them are going into the slurp jar?
No, just the crinoid.
Mike,
So-
can you open up the bottle?
Okay.
Then we'll sample a
Bottles.
piece of that coral.
So 2 samples then?
Cans.
Yes.
Okay.
The cans, yes.
Switch cameras.
Yeah, cameras.
Look at that. Fanny.
Okay.
Nice.
That is
okay.
insane.
On 6.
Yep, on 6, you want to...
So we have word from
do have,
Megan that the
ability to
fish
rotate,
earlier was a
adjust
sleek
if
head.
Sleephead.
Sleep, like night sleep?
Sleek.
Slick.
Sleek head?
Slick. I don't know,
Slick head.
whatever in English.
Slick. Like slippery, I think.
Look, yep. That's the one right there.
Slick head. Oh, I love that fish so much.
Wow. That is crazy.
It almost looked like it had like a robber's mask on. So that definition's pretty cool.
Slick
Yeah.
head fish.
The shot of Herc right
Like
now is
oil.
pretty cool.
Yeah, the image of Hercules on channel 2 from Atalanta. Looks pretty awesome.
Hey, Mike, you think we could get a quick zoom on the cinema cam?
Yes. Yep. 1 second. Okay, zooming in.
That's perfect.
That look good?
Oh, yeah.
Roger.
Oh, yeah.
Oh yeah.
Thank you, sir.
No worries.
Oofta.
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 That's awesome.
Whoa, this is amazing.
Yeah. I don't want this shift to end. We only
I
have
know.
about 30-ish minutes left of at least our watch, and then the 8 to 12 crew is
Does
going to
that
take
door
over.
lock?
I know.
God.
Does the door lock? I'm with you, Benny. I'm with you on that one.
Oh, that's great.
Hey, man.
Let me know
If
when
it's
you're
unanimous.
ready for suction.
Okay.
What did vomit do to my jaws?
What?
Fanny, we have a question about how do we go about selecting what to sample or to not sample? For example, if there's a lot of things available versus just being one thing, and so.
Yeah, so that really depends. So for instance, here there's different things that we want to consider. So we want some sample that is representative of what is there. So by focusing, for instance, on what the most common type of organism we're right now seeing in that area can be highly valuable, especially to compare then to the eDNA that is being collected. There are also here some of the species that we're seeing are pretty common across the Pacific and have been sampled pretty widely, so this may not be a priority right now, and we'll be maybe focusing more on organisms that do not seem to have been sampled anywhere else in the Pacific Ocean or elsewhere. So that's a combination of what is representative and what kind of sample we already have available and what is rare. So there's just a lot of different things to consider.
Do you want a little bit of a zoom, Danny?
I need more suction.
More suction coming up. 73. You want more?
Oh, yeah, that's not pumping. It's not sucking. Remember, you've got to use the suck button on the... Turn off suction on there.
Suction's on.
Turn it off. No, on the GUI. Okay, now hit it on.
I have control of the GUI. PRV.
I understand, but the button on the GUI doesn't work.
Okay.
Come on.
Is it just attached now? I think it's really attached, so maybe we can just clip that piece with the coral on it, if with the crinoid on it.
Okay.
If it doesn't go, it's gonna have a hard time swimming now.
There's a piece of the crinoid in the...
Yeah, we got like a couple of pieces of the crinoid.
Suction going off.
We're collecting the whole thing now?
Well, not the whole coral, it's big. Like that area, maybe like
crinoid with
yeah.
the coral on it?
Let's see, maybe a little.
Or
Can we zoom in? I have a hard time. Like,
Zoom in video?
Yeah, something around like right below that. Yeah, somewhere around here.
So about where I am?
Yeah. I guess.
Changing to
nice
starboard bio box.
Starboard?
That's where you want it?
Yeah, we can put it in there.
If we can, try and put it in one of the smaller ones, if possible.
Oh, that's a large.
I know, but we want to try and save room for the rocks if we can.
Or we can put that in the forward bio box then, because I don't think it's going to fit in the small compartment.
Yeah, I agree.
Okay, forward bio box out.
Forward bio box. Okay. Forward bio box coming open.
And now when you go all the way out, bump it back in just a little bit.
Coming back in.
There you go.
Okay, retracting the camera.
Yep.
Camera's fully retracted.
So Hayden, 178 was the crinoid slurp and 179 was the coral grab.
Correct.
Thank you.
Nice.
All right, thank you.
Okay,
Good
pass
stuff,
it in.
Danny.
Okay, closing forward bio box. Forward bio box closed.
All right, so now let's continue looking around for other potential corals to sample.
So for our pilots, there's a question about controlling. When you're controlling the ROV, is there any delay between the input and when it actually happens?
No.
Pretty instantaneous.
Wow.
That's awesome.
Speed of light.
I see a
Ooh,
neurinovorgia.
yeah, that's a big one down there.
We can zoom in on it. I don't think I'm going to collect it, but we can have a zoom on it.
What do you want to zoom in on?
Let me show you right here.
So whoever asked about Paragorgia earlier in the chat.
Do we know which slurp jar we sucked some of that crinoid up into?
Number one.
Yeah.
Number 1? Thank you.
Everyone's wishes are coming true on this dive.
Where'd that squid at?
Where that squid at?
Some patience, it'll show up.
Right?
You saw it at the beginning.
Yeah, we
Yeah.
did see a little squid at the beginning.
That is
You
really
didn't
cool.
mention what size squid you
That
wanted.
is true. Yes, yes.
Be more specific.
All right, now let's ask for some big squid. Come on.
Somebody says, you guys make me want to do a career change.
Brandon,
Do
you got
it.
Video?
Join
it.
us.
Do it.
It's never
So
too
pretty,
late.
these
Possible, man.
corals.
It's possible.
Wow, that is amazing.
Beautiful. So we can definitely see why it's also called a firework coral. Just stunning.
You're on the other side of a wall from me. Just as a heads up.
They just never get old.
No, they really don't.
Yeah, there's another sort of like overhang / cave also below. All
Okay, go
right,
wide
zooming
video.
out.
Going wide.
So I would just maybe circle back to that large structure behind us where there was the crevasse, and on top of it there was a lot of different corals.
Yep.
This might be a spot for sampling as well. That's beautiful.
That's amazing. You don't get to see this every day.
Oh,
Nope.
no.
Unless you're a video annotator, and then you literally see this every day.
Looking at you, Megan. People in the chat are also manifesting what they want to see. We have megalodon in all caps. I got it.
Let's
Somebody
get it all.
wants somebody
Let's
wants
get
to
it
see
all.
a vampire squid.
Oh yeah, that
Oh,
would be
yeah.
nice.
Incredible.
I agree with that person.
Also, we have a 4-year-old named Easton watching.
Hi, Easton.
Hi, Easton.
Hello,
Hi,
Easton.
How's it going?
So cute.
I hope you're enjoying it.
Yeah, lots to explore down here.
It's insane. Look at that.
There's
This
All the
is
sponges.
so incredible.
Wow.
Good gosh. Goodness.
So excited that this is a marine sanctuary now.
Oh, yeah.
How
Yeah.
exciting is that?
Yeah.
Rightfully
Yeah, look
so.
at the size of these sponges as well.
Jeez.
It's insane.
Solid dive.
That
And
dang.
we just barely got here. Like
I know.
We
the next
just
shift
got
is
here.
stealing it. I'm joking.
we can share. We can share.
I know. I don't wanna.
I know.
I can't get over this picture, man.
So amazing.
I can't get over
All of it. Everything.
the walls. Like it's just filled to the brim with life.
Mm-hmm. Oh, man.
It's unimaginable. Like I can't imagine how far on this goes for. You know, we're just seeing like maybe what 1%.
1% of it.
You know.
Yeah, just the little bit that is illuminated by the light from Hercules. That is what we are seeing, and the rest of it is
Pitch
in
black.
darkness.
Just
Yeah, it's like a car
unimaginable.
with headlights
Can we
on the back.
move the Zeus cam a bit up so we don't see the porch in the frame? That, thank you.
Thank you.
Oh my gosh, what a view.
The whole dive
Mm-hmm.
is spectacular.
Really is a lot of bamboo corals on here.
Is that the one Louisa was most excited to see at the in her talk?
What is
Nothing
the
behind
question?
me, right?
Nothing's behind
Nothing.
you. That I can see from their aft light.
The sonar looks clear.
Seems like we have maybe a brisingid here.
Oh, that's a first. Haven't seen that one.
Look at that. Look at as you like look down on the other camera on the left.
Mm-hmm.
Like it just like disappears. It's whoa.
Just keeps going.
Jeez.
Somebody
video?
said,
Zooming.
good luck choosing what to sample on this one.
That's
It's
fair.
also tricky because there's a lot of corals everywhere and we
Yeah.
don't want to destroy
Of
like
course.
other corals as we're sampling. So
course.
actually choosing a sampling target is tricky.
Yep.
Mhm.
Is that a sponge? That looks like a antlers. Or is that
Maybe it
coral?
was-
What do you
I don't
think?
know. I think that was from a coral.
Oh, I see. It's so white.
Go wide video.
Oh, there it is. See. Yeah, we can keep going.
Jeez, I looked over and that, I don't know what that is, that really white thing on top. I thought it was like a bat flying at us.
I know, that one
Oh.
got me too. It's just like some sediment that collected in a rock in a very specific shape, it looks like.
Look at that view from Atalanta, though. That is insane.
Yeah. All right, let's go around, continue going around this rock.
Somebody wants to know if we have ever found brain coral that is this deep. Is there like a deep water species that we know about?
No, not that I'm aware of. These are mostly shallow water scaractinians.
Uh-huh.
I'm not aware of any deep sea brain coral. We do have in the deep sea is Caractinian corals that form reefs as well, but they're different, a little bit different.
Okay.
There's more sponges here.
Whoa. Is that another sponge right there?
Yeah.
Okay.
Here?
It's massive.
Yeah. Or that circular looking one.
That
Oh,
one here.
something swimming down below.
Oh yeah, in the bottom left.
That's a big boy.
Some sort of eel or something.
Yeah.
Starting to come up tight.
How about here? I see like some shadow of a coral that look a little bit different.
How's my tether looking?
You're just coming up tight. I'm at a 17-meter altitude. So if you're gonna go down, we might have to move.
Yeah, I'm gonna go down a little bit.
Okay.
You want to look at this coral hanging out over here?
I can do like a 5-meter step towards Herc.
Sure.
Okay.
Yep, I'm getting pulled off.
Zoom in video.
Zooming.
Okay, can you zoom in?
Oh, that's a beautiful coral right there.
Dragging me.
Yeah.
Oh my gosh.
I'm stretched tight.
We'll get back to it. I just gotta get out of line moving.
That's all right.
Fanny, do you have any insight as to how quickly these deep sea corals grow? Somebody specifically wants to know about bamboo coral, but I guess it would depend on the species, right?
It depends on the species, it depends on the location. In general, deep sea corals grow pretty slow. And as you go deeper, they are going to grow slower, mostly due to their slow metabolism, I guess, and low amount of food available. But we know that some corals can be over hundreds of years old. Sometimes some of them have been aged to be over 1,000 years old. So they are very slow growing organisms.
It's just so interesting to think about, like, potentially the coral that we're seeing right now could be hundreds, if not thousands of years old. Again, we don't know for sure with the coral that we're seeing right this second, but it's just really interesting to think about.
And then you know how I feel when I accidentally knock one over, or I'm like, wow, I just killed a piece of history.
Well, you don't mean to, and it doesn't happen very often. You're trying your best. I just saw a couple of little floaty jellies on the channel 1, so we had a couple of tiny ones there.
More jellies, please.
More jellies.
It's starting to come up tight again.
Do you want a second step?
Yeah.
And we have Caroline in the UK listening to us, just being delighted about this seamount. Thank you, Caroline.
Still over
Shout out Caroline.
Shout out Caroline.
Yeah.
It's hard to not be excited. I mean, it's just...
Yeah. I'm going to be sitting in that lounge watching the whole time.
Uh-huh.
There's so much marine snow. I mean, look at that.
So much life.
Yeah. I think, you know, seeing like flat substrates really cool, but like seeing so much diversity of substrate and blue water really ups the ante on that tension of seeing something. Personally.
I'm right there with you, man. It's crazy.
So I am doing my Instagram takeover today, and somebody is saying on the chat that they expect an interpretive dance as part of my takeover.
Oh.
I can let you know right now that is not going to happen.
I think part of your takeover should be learning cribbage.
Oh, you know what? Yeah.
You want to do it?
Yeah. Sure, I'll do that one.
Yeah.
Right.
Okay.
Interpretive dance, no. Cribbage, sure.
Just one game. Yeah.
Yeah.
It's no pressure.
Are you getting any relief pilots?
You're stretching out again.
Why can't I see anything?
I think it's to your starboard and down. I think you started dragging ahead a little bit. And yeah, I'm at 25-meter delta, but only 14-meter altitude. And wall looks like it's about
So much
maybe
snow.
about 15 meters off to your starboard.
Yeah.
Wow.
Okay, start
This
to
marine,
see you again.
this marine snow is just crazy.
Mm-hmm.
Okay.
That's a great sign.
The only snow we have here in the Marshalls.
It's true.
Oh, look at that iridogorgia down there.
I think we're gonna do watch change, you guys.
Okay.
Sounds good.
I don't know if that sounds good, but
Yeah.
Yeah, good point.
Lock the door.
Yeah.
We'll be back in 8 hours.
Yeah.
Will we? Because it's a 12-hour dive.
Yeah,
so
I think,
Oh.
yeah.
You're right.
we
Math.
shan't.
Oh. You never know.
I feel like, isn't it coming back up during, no?
I think we should stay
I
on the
don't know.
station for longer.
Is that a mushroom coral on the bottom right?
there's a few of them. I don't see it right now, but very likely.
So amazing. Yeah,
That
we.
site is, oh yeah, that is a very nice
Okay.
mushroom
it.
coral.
Uh-huh.
Hold and stay
Over the
close to
gorge.
10-meter altitude, so you have a little bit of slack if you want to move. But
Took 2 of those steps, so hopefully it'll start feeling more and more
Come on,
comfortable.
Robert. I need a bio break.
Yeah.
You see those floating around? It look like. Does not look like snow on the top right over there.
No, I see what you're saying.
Uh-huh.
Perhaps
You
it's
see
just
that?
perhaps it's just a close view.
Ah.
Might be distorting it.
Could be, yeah. Yeah, yeah, yeah.
So it is approaching that time for us to do our watch change. So we are the 4 to 8 crew. hanging out with you just for the next couple of minutes or so before the 8 to 12 crew takes over. But once again, you are still certainly welcome to hang out, join the exploration, give us your questions, your comments. We really love to see them. But if you start hearing other voices here in just a couple of minutes, that is what's going on.
Certainly has been an honor.
Oh my gosh, so much fun.
Go ahead and zoom video.
So dark.
That's such a dramatic view of like the stark contrast of.
There's a shrimp in that
abyss.
Sponge, dead sponge.
Oh yeah.
Stock.
The glowing eyes.
It looks like a primnoid coral here. That's not a bamboo.
Thought I saw lava. Guess not.
I saw Eli doing the winch checks. So.
So much happening.
Yeah, it looks like you have a few like corals covered in zoanthids
Go
as
wide
well.
video.
Going wide.
There really is a lot of both density but also diversity at this spot.
Right.
Yeah. I think you're right about that current. Based on the marine snow that we were seeing up there, like there's definitely a reason based on that current. I can't imagine how this would look like the whole thing.
No, it'd be really cool to orbit this.
I agree.
What does that mean?
Alrighty, Danny.
Okay, Danny's signing out.
Danny's signing out.
Bye, Danny.
Norbit is a multi-beam that we can mount on the brow of Hercules.
Okay.
And it gives us a lot more finer detail from being so close to the substrate versus trying to map with a multi-beam from the ship.
I see. So just like a more detailed view.
More detailed view. You can get a whole lot more information coming from the Norbit, and then we can also mount it instead of a downwards looking position. We can put it in a forward looking position, and you can actually kind of see water column data from that as well.
Oh, very cool.
Yeah. But really, really helpful for creating three-dimensional models of areas and just getting a lot better viewpoint of what's actually going on.
Gosh, it's so tense. I just want something to come up from the bottom.
I know, I keep watching the darkness.
Oh, I know.
Megalodon.
We haven't gone past waypoint 1, just here.
Oh man, so much left to explore.
So much.
Dive extension.
This will be a cool one.
So yeah, we did a couple like loops. You can see where the samples were. We kind of carved around this piece, a second piece, and now we're drifting over here on this wall that you have on your starboard side. Yeah, we're not even, our waypoints are actually northwest, but we're, seeing lots of fun coral garden-esque things. Yeah, much more than rocks, actually, much more. Tons of coral.
Could you call it like a coral farm?
Ooh, coral farm smart.
Like, I mean, it's not like you're farming it, but I feel like there's an alternative word here we could find.
Coral. Coral collection? Nah. Someone's asking, how many cameras are located on Herc? If I remember correctly.
I think it's 16.
I think it was like 12, right?
Oh, on Herc alone.
Oh, I was going to say 6. Oh, man.
On Herc, I think there's 10.
10?
10 or 11, because they have all the sample cams and everything, too. So
Keone signing out.
Bye, Keone.
See you guys at breakfast.
Yeah, I definitely heard the answer before, and I completely forgot it.
I can't remember.
Yeah, see if the next oncoming 8 to 12 pilots might be able to answer that one.
Bye, Brittany.
Stand by. Bye. All right, thank you, everybody, for joining us on our 8 to 12, excuse me, 4 to 8. 8 to 12 is joining right now, and we'll see you on the next dive. Thank you.
No, they're going that way, and we're supposed to be going that way. They've done this. Yeah. Yeah. Look, that looks more tasty up there, right? Oh, we forgot to have a discussion about tether wraps. I think I have to have a +1 in. When does this dive end? Oh, somebody else's problem. There's a big fish. Yeah. Can we kind of scooch off of the wall here? So we're not. Yeah. Yeah, you could make one move that direction anyway. Just to get us off the face.
Hey there, back row.
Hey.
So we're thinking maybe we should actually start heading to our first waypoint instead of away from it. What do you think?
Oh. I know. Wait. I was like, Val just said, well, Val's back.
Okay.
I was like, I don't know if I can make that call.
Oh, I thought she was sitting down. Sorry.
Yeah. I was
yeah,
like,
oh, this works. Oh, no. I was like, on the eDNA front, I think that works. I've got things paused until we move again. So,
ooh,
yeah.
okay. How exciting.
So did you put in a move?
I did put in a move.
Okay. That way?
We've
Yeah,
got something
that way.
swimming up there.
Okay.
It's just bursting with life here. This is crazy.
Yeah, I lost a ring, correct? Yep. Shout out to my mother who let me know about that very early this morning.
Okay. I'm getting
Okay.
off the wall.
Yeah, no more wall. Hey, Val.
Hey,
We made
yes?
an executive decision up here and decided that we would start heading to waypoint 2 instead of away from it. How's that
Heading
sound?
toward it. I have no objection to that, and I would have requested the same thing. So
Okay.
back row has no objection there.
Great.
Yeah, I apologize. I had to go grab a charger.
It's all good. We just realized that we had gone the opposite direction from what the dive plan was. And it's been really cool over here, but I have a good feeling about this
Me
dive
too.
plan, and I think we'll find even cooler things if we keep going.
Likewise, and I agree with you on all of those points. So yeah, let's get back on course.
Okay.
Although I do understand why we were off course.
I mean, this is a really cool feature. It's
It
got a lot
is.
going on.
Yes.
We've been on bottom for almost an hour and a half, and
We've been on bottom almost an hour and a half?
yeah.
Okay. Well, I was asleep.
Yeah, so was I. I checked the log.
Wow.
We landed on bottom at 1838.
Okay. Yeah, okay. Yeah, let's get moving then.
I mean,
love
it's really
love
cool. Look at it. It's gorgeous.
I love this footage though. I've been
incredible.
waiting for this all
This is
expedition.
beautiful.
Yeah.
We can do it tourist mode here.
Yeah. We're just moseying along.
Enjoying the view. Wow.
All right, looks like you're a little zoomed in there, Levi's. I think Mike left it that way for you, so just letting you know you aren't.
Just to add some excitement.
Yeah.
More drama.
And this is what we have in the sanctuary.
Wow.
Yeah.
I am here for it.
That's incredible.
All right, I'm at the end of the rope. We can look at stuff.
Yeah, last night
Wanna
Hayden
zoom
was
in?
asking me what
zooming.
would be like the coolest thing that we could see on this dive today, and I was like, super high dense coral community with a lot of like amazing associates and things like that.
So
Here
you
you are.
manifested
Ta-da!
this.
Manifested.
Oh, wow.
So we're seeing lots of bamboo corals, crysogorgid corals. There's quite a couple different kinds of crysogorgids. There's crysogorgia and then Ramillogorgia militaris. And there's some like planar looking colonies as well as bushy looking colonies.
Oh, I'm extra bouncy.
There's zoanthids overgrowing a bunch of these corals as well.
All right, so I'm trying to catch up with the science chat. It looks like we need to keep eyes out for potential collection for a Hemicorallium, any scleractinians, primnoids, and I believe we will have eyes on the stream helping us out with that too.
Okay.
It's quite the shopping
So
list.
now that we're moving back on course, I'm going to resume our eDNA. So Vale, can you Hayden had indicated that pumps 0 and 1 were paused.
All right.
Okay.
So I just want to make a note that we are resuming them at this time.
So you're resuming
Pump
pumps
0 and
0
0
and
and
1.
1.
Okay.
So, okay, so 0 is going. I'm turning on 1. Okay, they're both going. Can you tell me the depth? I like to make a note of that in my notes as well.
Current Herc depth is 20 meters.
2020.
Yeah, there's so many corals here, you almost can't see the rocks.
Yep. Well, that's a problem when you want to rock. No, there's nothing we can grab here. So the corals can and should be coraling away.
This is a really nice spot.
It is.
Probably lots of current acceleration right here on the wall, which makes it perfect for these corals to find food. And obviously, you know, the red's probably really high around here. It's an ideal spot. There's also a bunch of hydroids, lots of associates. I'm seeing brittle stars and crinoids and squat lobsters.
Wow. Whoa.
Oh, big
nice
sponge.
Big polyopogon sponges. Yeah, those
about
polyopogons
time.
can get huge.
The polyopogons do get huge. We've seen some that get as tall as the ROV in some areas.
Really?
Yeah.
Oh my gosh. It's massive.
They can get enormous.
Look at this.
I'm a joy gain cranked. I hate that.
Some of my favorites are those giant bolosomas you can get in the in parts of the Northwest Hawaiian Islands with the bright yellow stalks.
Oh, yeah.
Yeah, some of the ROV pilots will call them cheese moon wheels.
Yeah. Cheese moon wheels.
It makes me very happy.
A lot of times they're like full of brittle stars.
They are, yeah.
We might see some of those.
I didn't know if we were in the right range for those or not.
Yeah, you could see them down here.
Okay.
It might be a little shallow. But every seamount is different. You never know what you're going to get.
Whoa. It's incredible.
It's amazing. Whoever has to annotate this dive is going to have a fun time.
Let me guess. That's going to be you.
Likely.
When do you encounter something like this when you're annotating? Like, what's your process? Because it's so dense.
I try to go and count everything in a frame. And when there's high density, we do categorical abundances instead of, you know, just like straight up counting numbers because it's overwhelming. So over 20 animals, we'll start doing groups like 25 to 50, 50 to 100, or over 100 individuals. And I'll recount after we've sort of left an area, and I do that by, you know, we've moved the ROVs so nothing of the previous frame is in frame anymore.
Got it, yeah.
It takes a long time. Oh, there's one of those urchins that we tried to collect.
Oh, the green urchin. Do we want to try that collection again?
I don't know.
Okay.
You see that right there, bottom corner, the yellow greenish thing? Yeah.
Right there?
Yeah.
Yeah, that. Oop.
That might be really hard,
Okay.
just given that we're on a cliff.
And given what you've described to me about this species, it's probably not going to go very well.
No, it's super fragile, and we would have to suction it. And
all right, let's pass on that then, and we will focus on looking for some of the priority species.
We know it's here.
Yeah. No, we can focus on the priority corals that were discussed in chat. So, yeah.
Yeah, this is a little alcove here, so it's pretty.
Right.
I need to get out of this alcove.
Yeah.
Yeah.
It's a little dangerous.
Yeah, let's not get trapped in there.
It's cool. The corals like it, but you know.
Herc doesn't.
Our robots don't like it. Wow. This is incredible. Oh my gosh.
Yeah. This is definitely a high density community. I think there's a Hemicorallium right there.
One that's kind of side-on?
Yeah, that we're like looking straight down at.
You want to look at that?
Yeah, let's look at it. Since it's on the list of desires.
It's not an impossible task. We'll probably move it.
Yeah, I could stop the move if this is something we need to collect, but we will see another one.
Is this the Hemicorallium?
That's a Hemicorallium.
Excellent.
But it's a side-on corallium.
Yeah.
Yeah, that's a tricky angle to work.
Well, it's not tricky to grab. It's tricky to image.
Yeah, exactly.
You can maybe get under it and look up at it.
Oh.
Sorry, I just had my mic sticking out because I was coughing.
Hello again.
Okay, green urchins are on the request list from the shore team.
Okay.
Look up at it.
Look up. There
I have a little list going back here.
Is it slowly getting bigger the more we see?
We're just up to 4 items. That's not bad at all.
Yeah. Oh, okay.
That is very manageable.
Sounds good to me.
You can zoom in.
All right.
Oh, look at that shot.
Oh my gosh.
That's dramatic.
that's
Beautiful.
Wow.
I vote for this for the main promo image for the sanctuary.
Yep.
Yeah. Oh my gosh.
Hemicoralliums always do make for great imagery. They're really beautiful corals.
Look at that.
That's a nice thick base. This is a very old colony, a few hundred years old.
Wow.
Wow.
Oh my gosh.
Oh,
Oh, yeah.
close.
They're very fragile. They're really hard to collect.
Mm-hmm. Yeah, they just tend to break apart.
Wow.
I was annotating a dive where a fish got startled and just like swam right through one and it just shattered it.
Oh
into
my
a
gosh.
whole
What?
Yeah.
Oh.
So what's the deal here? Are we trying to get it?
Do we want to get it back row?
Could we
move?
slurp some of that branch that's hanging down?
Yeah, that little dangly bit.
Broke
That-
a little bit. We could slurp the brokey bit. I think I got a hover though.
I think that might be the easiest way to do it at this site.
Yes.
Coming wide on camera.
Wow.
Yeah, short team agrees. If we can get that piece somehow, they'd be very happy.
Just gonna fly the suction sampler right up to it.
That's kind of hazardous
Yeah.
since it's so fragile. I don't know, but I don't really have a great perch here either.
Right, there's something to land on. Oh, I can turn this section on. Do we have a jar already?
Can you switch over?
Jars 3, 4, 5, and 6 are available.
Okay, can we go off the flush jar?
Yep.
How much leash I got?
What happened?
I got leash. We're heaving a little bit. I mean, maybe we could fly the suction.
Okay.
I mean,
Do
otherwise,
we have beans on the suction?
can you point down a little bit more?
Okay.
And can I get more iris? More zoom? More camera? More zoom, more camera? You got suction?
Yep, suction's on.
Zoom and camera. We're gonna get out of the. Can you rack the camera back to here? Or we're okay. All right. Let's see. Which is the loose bit? Is it the hang down one?
That
right
little
there?
hanging
This
down
one right
bit.
here.
Come on. Oh, Bessie. Can you give me a little more leash?
Yep.
Well, there it goes.
Oh.
Oh.
That's a start.
Yeah, that was amazing.
It's not in the box.
No,
But we have it.
Just getting it like that, that was really impressive.
That is impressive.
All right, starboard bug, or forward bugs coming out.
Oh, we're putting it in a box?
We're going to put it in the box?
Put the box
Yes.
back in. I gotta pick it up.
Oh. Well, it's not going through the hose like that, so.
No.
Not without
encouragement.
a nudge. All right. Zoom. We're putting it in a box, right?
Yeah.
So we decided. Oh, let me adjust the grip force. All right, box out.
Box out.
Oh, don't you be dare do that. Which box they going in?
I think you can put it with
Lambda?
the bamboo coral
The
as
Omega.
well.
Which one?
Omega?
Omega.
Omega.
Dang it. All right. Suction off.
Section off.
It's stuck.
Get off.
It'll go. It'll go.
There it goes.
Nice job. That was
Yeah.
awesome.
Great. Awesome.
All right. I can put a check mark next to the Hemicorallium.
And is that 179?
180.
180.
All right.
Nicely done.
Onward and upward.
Yeah.
All right, let's do it.
Wow.
So one of the things that seems to be really helping with the population density around here is you can see from the marine snow that there's a bit of current that we haven't seen in during previous dives.
So the density is based on mostly on the current?
Not necessarily, but it is one of the factors. It just means that you have a better chance of accessing nutrients when you're fixed to a wall. Yeah, when you're fixed to a wall and you're filter feeding.
To the side view camera instead of the bucket.
Yeah, you don't have a ton of choice in what you get to eat.
Is that the sample bucket?
Yeah.
Go back to the bucket, put it on the slurp. Or I mean this flush, sorry. What am I talking? Flush, the red one. There you go. What's in there?
There's a piece of bamboo coral. I think that's from their slurp adventure during breakfast.
Why don't you run the slurp for a second?
Okay.
It's probably good.
Whoa.
What an amazing feature.
Yes.
Yeah, so with the whole current thing, think of it kind of like if you're standing under a tree in your yard with your mouth open. If the wind's blowing, you're more likely to get a leaf in your mouth rather than just waiting for it to fall.
I like that.
But you also, if you want a leaf in your mouth, you have to be near a tree.
This makes sense. This makes sense. I like this.
Oh, there's water.
Dusty.
Here.
Oh yeah, you can see it.
Yeah.
Wow. Got sucked into the crevasse.
Void.
It calls to you.
Danger.
You're like, oh, it's so beautiful.
Yeah, let's stay out of crevices, please.
Very tempting. Wow. That's incredible.
Okay, now I'll switch over to that other camera view.
Okay.
So I can see what's going on over there.
Yeah.
All right.
Wow. I think now would be a good time for introductions, by any chance?
Oh, sure.
Yeah? All right, cool. I guess I'll get us started off. Hi, everybody. For everyone tuning in, my name is Giselle De Leon. I am a Science Communication Fellow with EV Nautilus. When I'm not on EV Nautilus, I am a lead STEM educator at the San Antonio Museum of Science and Technology in San Antonio, Texas.
That's one of those predatory jellies. The red thing there, yeah. All right, so my name is Val Finlayson. I'm sitting in the science lead position. I'm appropriately one of the science co-leads on this expedition and the geologist on the ship. And back at home, I am an assistant research scientist at the University of Maryland in College Park. I do high temperature isotope geochemistry on rocks that we collect on these kinds of expeditions to try to understand how Earth's interior works.
Hello everybody, I'm Luisa Dueñas from the National University of Colombia. I'm here also with Ocean Census. I'm in Science 2 SIT, and I'm a biologist helping with all the cool collections, sample collections we have.
Good morning, everyone. My name is Jovel Vrane Dako from the University of the South Pacific. I'm an ocean science intern serving as a data logger.
In your front row, I'm Megan Putz. I'm the navigator on this watch. I'm a deep sea biologist at the University of Hawaii, and I'm really excited about this dive today because of all these really cool animals we're seeing. It's going to be great.
It's very cool.
Yeah, I'm Robert Waters. I'm a Herc pilot on this watch, and I'm OET facility manager and ROV engineer working on electronics.
I'm Levi Zabawit. I'm from Columbia University. I'm on the Atalanta seat, and I'm the ROV engineering intern.
Lots of buttons over here. Hi, I'm Amber Flynn, lead video engineer, and when I'm not on the boat, then I am a cinematographer and camera operator based in Los Angeles.
I'm Annette Govindarajan. I'm a biologist at the Woods Hole Oceanographic Institution. I'm one of the science co-leads, and I'm conducting eDNA sampling using autonomous sampler and Niskin bottles to look at the amazing diversity of species that we're seeing.
Oh, thank you, everybody.
So at the moment we are working in the southern half of the Bekar and Bokak Marine Sanctuary, newly established in the Marshall Islands. We are on bottom. We're just a little over 2,000 meters down, and we are moving toward waypoint 2 on our dive track.
So what features helped y'all decide
Zoom in here.
this route that we're taking?
Zoom out.
What were the features that helped the team decide this is the route we were going to take, these waypoints?
Out. I want to see what we're doing here. Okay. So that's a ridge, sort of.
Yeah, we're on a ridge. Okay. Yeah, here's the map going up this ridge.
Okay. So we were doing some mapping late last night, and the science team got up very, very early. We had a 3:30 a.m. meeting today to decide, based on the new map data, since this feature hasn't been mapped before, where we're going to dive. And it was pretty obvious on this one because this is a pretty steep-sided geo, pretty large volcanic structure. And we mapped one really prominent ridge over on its southeast end, and all of us were like, yeah, that's where we're going. So we just had to figure out what the depth range and length of the track was going to be, and that always takes a few more minutes to hone in.
Yeah.
Because one of the things we really try to do when we set up those tracks is once we have, once we agree on a feature that we want to dive on, we have to figure out how to best balance those objectives that we want to meet scientifically.
Yeah, a little bit.
So
this is skosh.
Okay.
One skosh.
So we want to move backwards just a little bit. Because it looks like the wall is.
Well, I'm facing.
Wow. You're facing 154, and we're going 300.
I'm sort of at the wall. I'm sort of facing at it.
Okay. So.
I mean, it is. That's the right slope. It's just we just need to get just a little bit over there.
Yeah, we just want to move up here. Okay.
Yeah, just a little off of it because there's kind of. I'd rather it was behind Herc instead of.
How about I make
track.
a 20-meter move north?
Yeah. Okay. I like it.
Okay.
You already have a move in, like along that track.
Yeah.
Maybe just skosh it a few 5 degrees or whatever.
Okie dokie. I'll change it to 320.
Okay.
Bridge now. Can we make a 20-meter move 320?
So yeah, with all of the different science objectives that we're trying to achieve on this cruise, it's important to always have that conversation before every dive planning meeting to make sure that we can take care of as many of those as we want to for the team, as many as possible. And we can only see so much in the map data. There's only so much resolution that we can get when we're looking at something, 2, 3, 4 kilometers below us. That's a lot of distance there for sound waves to propagate and come back to the ship. So one thing we don't really have a good idea of until we're on these features is what this fine meter scale topography looks like that we have to navigate. And I think what we're running into right now is a case of a lot more little local vertical steps along the bathymetric profile we're surveying than what we initially expected from the data. We know we have a steep step coming much further on in the dive, about halfway through the dive track. But yeah, you don't know if you're going to encounter little rock walls like this along other portions of the track until you're in them. So things like this, we figure out how to navigate when we get there.
Yeah, sometimes these features, they're hard to see on the map because they don't happen over a long distance, and so it kind of blends out. Ooh, a gillocrinus. Is that the stalked crinoid?
Zoom in?
Yeah, let's take a look.
These are the ones with the anchor?
Yeah.
Yeah,
so
this one here.
Look at that. They remind me of sparklers.
Yes. I agree.
And all these tiny romilogorgias.
Yeah, those are everywhere.
Yeah, it would be like categorical abundance 100 plus. So many of those.
Wow.
I've seen at least 3 different kinds of bamboo coral. Maybe four different kinds. At least. There's a lot. There's a lot of kinds.
Yeah, there's a lot here.
Up. Telestrator computer is attempting to restart.
Oh, don't do that.
All right, I'll take a peek.
I've paused it. I just need to bring back.
I'm just going to do a test.
Okay. I think it is just running a software update or attempting to.
Okay. Looks like I have control here.
Okay.
But is it opening for you again?
Let me double check.
Otherwise I can come back.
Yeah. I don't want arrow. There we go. Yeah, it didn't close anything. It just minimized the window on me.
Okay.
So it did this earlier in the expedition too. So we'll just have to run the update at some point.
This is crazy.
Yep, back row unable to draw on things due to computer update.
Is it back now though? You can do it or no? Can you draw now?
Yeah.
Yes.
Okay.
Yep, we are good to go.
Okay.
Big check mark.
Check mark. I'm surprised you didn't do a smiley face.
I try not to clutter it up too much. Because yeah, we have to use that too. That does go out to the audience. Also, at some point, the ROV pilots will tell you to stop, which they absolutely should.
Yes, this is true. Not too much tomfoolery.
I remember on past expeditions when the telestrator was still a lot newer. There were some emojis.
It was more fun.
It was more fun. There were emojis, and I remember an incident involving completely covering the Hercam view with a bunch of whales.
Oh no!
And
rascals.
a stop was put to that. I was not involved with this. I just heard about it from folks once I got on watch, and I noticed that there is not an emoji option on the bottom of the telestrator anymore.
Revoked. No more emojis.
Back row privileges have to be limited because sometimes we're like children in a candy shop around here.
This is true. I mean, if I could do a whale emoji, I would. All right.
Yes,
which is why it's not there.
Yeah.
You can still draw a happy
We
face.
pace
You could
ourselves
draw a
with
whale.
Atalanta, we could.
I'm not going to push my privileges back here.
There's a lot to see, so we can go slower and just look at stuff.
Right.
That's why you're in this.
Trying to make up time here.
That control.
Yeah.
I'm not in control. I'm just the manager.
We just have to like not stop and smell all of the flowers.
I mean the control not to do whale emojis.
But we can still smell some flowers.
We can smell some
We
flowers.
gotta get up there.
I know we gotta get up there. Bridge now. Can we make a 20-meter move, 310?
Yeah, I vote for some maneuvers and then some flower smelling.
Yeah.
Flower
It's a balance.
Yeah.
So was that same off? You're going more this way?
Yeah, I'm going more this way. Because they're still really close to the wall.
Yeah, I think Steve really wants to get up on the
the big
on top
stuff.
of the tallest, the highest terrace on this seamount. So, yeah, that's always tougher to do on dives where we do see a lot of biodiversity and a lot of high density communities like this one. So we'll do the best we can.
Maybe I should pull it further.
The temptation.
And we've been kind of breezing through the other ones because they've been so sparse.
I think maybe 340. Bridge now. Hi, can we change our bearing to 340? Thanks.
That'll help with the tail wagging too.
Yeah. The waypoints may be suggestions, but they are a general direction.
We could go up some.
Yeah, we can go
Yeah,
up
let's
some.
go up.
Yeah, a little up.
I like up.
All right, going up.
Going up.
What's this little pink friend over there?
Pink friend. Can we zoom on the
yeah.
pink friend?
Let's do it.
Pink friend. Ooh.
Ooh, that's spicy. I think it's a Brasingid.
Pretty color.
Yeah.
Is it just hanging on by one leg?
It's like hanging on a little old stalk of a coral.
Whoa.
Okay.
At first I thought it was one of those colorful
yeah,
shrimps.
I can see the skeleton now. It looked like a leg.
Yeah.
Look at that.
Yeah, it's got some white coloration that I'm not used to seeing.
Well, you
just
usually don't
frequently.
see the bottom of it.
Yeah, yeah.
Generally they're on the rocks, but this one's hanging out on this dead coral.
Yeah, funny.
I was a little confused. I'm like, what are you? Really
Yeah.
weird crinoid? A
Right?
Brasingid? Something I don't
All
know?
right, we good?
Yeah, that's good. Thanks.
Going up.
Going on up.
Goodbye, little friend.
This is so weird. It's like the rocks have kind of hollowed out underneath the manganese crust in a few places.
Yeah.
Not sure what that's all about.
Yeah. Well, I wanted you to tell me that. I've seen this on other dives, and I've never really understood what I was looking at. And that made me, like, question whether I was looking at basalt or not. But it's like, it looks like basalt.
I don't know. It looks like the crusts have really overgrown in a lot of places, too. So maybe they just have grown some void spaces. I've not quite seen it this hollow before, though, which has me a little worried that we might not be in basalt.
Right, okay. So it's not just me thinking like, oh, it looks so weird.
It definitely looks weird, and a different weird from yesterday. Like a more normal weird, if that makes sense.
Yeah, well, I definitely think, so like
on a weird scale, 1 to 10.
On carbonate caps, you'll see some of that like erosional features like this, and like you'll get these like little top hat situations.
Yeah, and that's what I'm concerned of, that I'm looking at here, because I was hoping we'd get some igneous rocks. But we'll grab a couple if we see an opportunity to grab some rocks, and we'll see what they open up and look like.
I guess this is the top of the wall feature.
Yeah.
That's pretty cool.
I was kind of...
Can we zoom like this little ledge?
Yep.
Yes.
Zoom.
Yeah, I was taking a gamble because the carbonate caps were documented to be pretty thin over on ODP site 144 over on Wotajibato Gyo, where pretty close to where we did our dive. So I was kind of hoping regionally that could be something I could rely on, but I might be wrong about that, because we are also further east, and some coeval seamounts over in the mid-packs, which we're slightly closer to here than at Wotajibato. Those have enormously thick carbonate caps, like several kilometer thick carbonate caps.
Really?
Yeah, we.
That's nuts.
So Lamont did a bunch of dredges out there in the 70s, and they got down to what they thought was hard rock based on reflection data.
Tiny pink thing on the rock. What is that?
So they dredged. They found a bunch of carbonate. We went back to a couple of those sites and dredged even deeper, and we still got carbonate.
Is that full zoom?
Carbonate on carbonate.
Oh.
It's just kilometers
it's
of carbonate.
All right, I'm good.
It's just there's some parts of the Cretaceous Pacific that generate these monster platforms.
Really good eyes.
I've seen enough of them.
She doesn't even wear glasses. It's incredible.
I do wear glasses. I just have my contacts in.
Oh, okay. Just superpowers.
We want to try and break off some thin bit of this to see what it is?
I don't think we need to collect anything off of the edge of this. That'll be all ferromanganese. There's all sorts of cool stuff growing on the underside, though.
Oh, yeah. Lots of, like, little stuff. The little stalked white thing.
Looks like a little mushroom.
Yeah, looks like a little mushroom. I'd be interested in seeing that.
Is it a sponge?
That's my max.
Oh.
Coming wider.
That's a hard one.
Getting
too tiny.
A little bit.
Yeah. Oh. Can I reset your DVL?
Yeah. But we're losing Doppler a lot. So.
Yeah.
Wow.
Can you tilt up a little bit so you can snag it? The full zoom?
All right. Got some more.
What is it? What are you? Oh, it's a carnivorous sponge.
Ooh.
Oh, those are interesting.
A carnivorous
You don't see one of those every day.
Yeah.
What? How does that work?
Kind of like a Venus flytrap situation, where small crustaceans like amphipods might land on it, and then it slowly engulfs the prey item and like eats it.
That's so sick.
But it's a sponge. So usually sponges are filter feeders, but in this case, sort of like how, most plants are photosynthesizers, but Venus flytraps will eat flies.
Wow.
Yeah, they're really wild. They're really cool.
That is so cool. I just learned that existed right now. That's amazing.
Widefield is giving us a really good preview of what we're going to see on top of this crusty layer thing.
Crust.
No.
No,
not crust.
Not crust.
I want what's under the crust.
Ooh.
And ideally under the carbonate.
Yeah.
Okay, moving on.
Okay, thank you. That was a really
neat.
weird looking thing.
Yeah. Your eyes are amazing.
Oh, wow.
Can we go look at the giant sponge?
Oh my god, what?
Wow.
Yeah. Can we take a little flyby of that?
What is that?
That is a giant sponge.
That is crazy.
All right.
I told you the preview looked good.
Yeah, but this looks like, whoa, like what? That doesn't even look real. This is insane.
Oh man, you need to go back and watch some of the old NA174 videos.
Yeah. I think I do.
We saw some of this on 138 and 154 too, just the whole monument area.
Yeah.
It's incredible.
Monuments seem to have these really massive sponges.
Look
They
at
sure
that.
do. That's why they're monuments. Save the sponges.
Save the sponges.
They look so squimpy.
Yeah. So this sort of leafy looking one, this one is called Tetroplural. And I'm still trying to figure out what this loby looking one is. It's different. Looks like a euretid of some kind.
That reminds me of some fungus.
So it's like more of a crunchy sponge.
Ooh.
The Tetroplural is more of a squishy.
Ooh. I wish I could like actually feel the difference between squishy and crunchy sponges.
Well,
Sometimes it's a little bit
go
of poking
downstairs and touch them.
Ooh, oh, then I'll take you up on that offer.
That looks good. I want to bite it.
Sponges are not very tasty.
No.
And
I
they're
think
very
I agree
silicate.
with you, Levi.
before because that's kind of what it tastes like.
I would bite it.
Your teeth would not be happy.
Is there like a little friend with legs on that sponge at like the bottom of the
Yeah, there's
screen?
a little
What is-
Munidopsis, the squat lobster, that little white.
Oh, look at that.
Little white one. And then there's a bunch of resinged sea stars and crinoids.
Wow, look at that.
On here.
Yeah, a blast. I want a little. That
Might be
nose
worth
fall off
taking
there.
a sample of the big lobie sponge.
I think so.
All right, we got
Let's
it
do
now.
it.
I'm gonna move in to get you closer.
Yeah, I think we can grab
Yeah,
a
get
pretty
out
good
of the
piece from on
tail
top.
wagging
Yeah,
mode.
yeah, exactly.
Okay.
I'm moving her up to you.
Okay.
Yeah, so we can probably come at it from the other direction from the top.
Yeah.
Yeah.
Well, there's an
'Cause there's a lot of it.
Anthomastus tahinotis.
Whoa. What is that?
So I'm gonna move the camera down to it.
Oh, that's a large one.
Yeah.
Yeah, right here.
There.
Ooh.
This is Anthomastus tahinotis.
So that's one of those soft corals again.
Oh, wow.
Oh, and there's long-armed spiny. Hope Steve's watching. Long-armed spiny. The observation of this swat lobster is something he had on his list. We have collected it. We just don't. He wants to note the presence of these animals. They seem to preferentially always hang out on hydrozoans.
Huh. Okay.
It's so precious. Look at those little arms.
Yeah.
Oh my gosh.
That's why I call it long-armed spiny. I knew it was unique.
Yep, Steve's watching. Munidopsis guachani.
Wow.
And it's
then-
recently described, so now it actually has a name and I don't have to call it long-armed spiny anymore. But I still like that moniker, so I still probably call it long-armed spiny just for nostalgia.
I love that.
And Tina says the Anthomastus is a tahinotis.
Yep.
Yeah.
Wow. Amazing. It's so incredible how much life you see, and we're so zoomed in, and you zoom out, and you're just like,
oh yeah,
look at this. Oh
you're
my
like,
gosh.
it's so everywhere, and then you zoom in, you're like, enhance, so many more animals.
More enhancing.
And then it usually gets weirder.
It gets weirder as you get closer.
Yep. Look at that.
All right, we can't get too close.
Yeah.
Yeah.
And the sponge and all that good
Then it'll
stuff.
get too interesting. There's a
Yeah.
Yeah, don't get too close to that sponge. It'll eat Herc.
Exactly.
Wait, it's not predatory, is it? No, I'm kidding.
Fortunately, the collateralized sponges don't get that big, so
Right.
you're not under threat.
Yeah, predatory sponges, I would imagine, are limited how big they can get just by the nature of the environment they live in and the availability of things to nibble on.
Okay, this is a little more conducive to getting in.
Yeah.
Could zoom in again.
This is crazy.
Yeah, my mind was blown when Steve told me that this was a Munidopsis.
Look at that. Oh my gosh.
Because most of the Munidopsis we see have short arms.
Oh.
This one has long arms.
They're like little miniature cranes or something,
I
just
know,
ready
right?
to pick up some little niblets to eat. Oh my gosh. It's so precious.
So Levi, our friend Asako in the shore team, saying probably another reason not to eat sponges is because the spicules and the glass needles are very, very painful.
Oh,
They get into places they should not.
My dream.
Interesting.
Interesting.
Yeah, some of them I've been told they're not very fun to handle in the lab.
As
Levi,
I would
do you
say,
still want
it's
to
not a
bite
good
it?
mouthfeel.
No.
Levi, would you still say you try it once?
That's good, yeah. I also saw, some like, looks like stoloniferous octocorals on the rock there too.
Oh.
Underneath.
All right, so we're done with that?
Yeah, we're done with that too.
Oh, goodbye, little friend.
It was really
so
good.
precious.
Lots of cool stuff.
Long arm spiny. That is a good name, though.
Yeah. Well, that's sort of what we do when we see a new animal. We know that it's unique, but we don't have a name for it yet. So we give it a placeholder name that will remember to be able to identify that same unique animal until it gets a name.
Aww,
and then we can replace that unique name with its official name in their database.
Yeah.
So we don't have to go back to each annotation and update it. It'll just globally update.
Oh, wow. That is so cool. And
Looks
very
like there's
efficient.
some big ectomyces off to the right too. That's pretty cool.
You want to take a peek?
Yeah, we're seeing a bunch of those.
Yeah, there's a lot of Tachynotus. Looks like there's a black coral right here.
Yeah, I think we've seen a couple of black corals here and there.
Yeah. This one's definitely going to be a hard one to work on. Gonna take a long time.
Can you zoom back out?
Okay, coming wide.
Okay. All right, I think we're...
Moving on?
Is it sponge time?
It's sponge time.
Sponge time. Sponge time.
Which
I think the best plan of action might be to suction a piece. It's gonna be kind of crunchy.
Okay, but which one? The one that's
the leafy one. Yeah.
Yeah, the big one.
The leafy one.
So I think we have done this. I think I remember doing this before in years prior. Chris Kelly was interested in this, and I believe that his advice was also suction.
Yeah.
Yeah.
All right, we're gonna do the fly-by-hose thing again.
Okay.
I need more iris, more camera, more tilt. Do that, walk it in. This is good. We're not getting the tail wag anymore.
Yep. Okay, it's not the tall one, but the...
Oh, not the tall one.
Not the tall one. The
one
big loby,
it?
yeah, the big loby one.
Oh. That's more Can we get it on the other side then?
Yeah,
it's the same sponge,
it's the same sponge. It's all the same, I think,
That'd
individual. Yeah.
We'll do that. That's better. This one's sticking out in her face. So.
I'm going to turn off pumps 0 and 1 from the multipuffer.
Okay, turning off 0 and 1. Are you turning on a new set?
One moment. Megan was asking if you turned on a new set of pumps.
No, not yet, because we're collecting.
Okay.
So is over here okay?
Yeah. Any of the loby parts is good.
Okay, can we zoom and tilt and do all that to get really close in?
Ooh, if you get the part with that little guy on there.
Right here?
Yeah, that thing. If you could get that part, that'd be great.
Right jar? We need jars and suctions and all the good stuff.
Yeah.
On it.
Which jar?
Think anyone but one, right? Vel? Yeah?
Yes.
Next one. One more time. I don't think that,
It's too good?
Go. That's just too good.
Okay, suction is on.
Okay.
Ooh.
Can you give me a little more leash?
Yep.
Yeah, you're still quite far away from it.
Yeah.
It might be easier to put the arm out.
Yeah. Yeah, I'm gonna get too close there. All right, we'll just go wide. You got a good Doppler lock? No.
No.
Of course not.
The nature of the cliff.
Can you give me a little zoom? Okay, more zoom. Tilty. Okay. I guess I need to drive in.
Yeah.
All right. We'll drive a hose out there. Can we zoom out some?
One more?
I think that's okay.
Okay. Levi's, I want to punch in a little bit on your Atalanta view. It's a good angle on perk.
Right.
So just letting you know.
Which lobe are we going for?
So you see that little sort of orange, pinkish thing on there? Yeah. I think that might be an aplacoforan, which is also on Steve's list.
What wagon? Tell me if I get too close to that overhanging one.
Okay.
Come on.
It's going to come into this.
Okay.
Let me turn a little bit.
Okay, so that'll be...
There you go.
Oh. I didn't get it.
Almost, though. That was the technique.
Come on.
Oh.
How are we doing with the overhang sponge?
It's okay.
Oh,
so close.
Yeah.
Go away. Sorry.
Who's calling?
Dang it.
It's probably spam.
Can we answer the phone and mess with them? This is the EV Nautilus. How may we help you today? This is your back row customer service agent.
Love some good mischief.
No, I'm sorry we can't do that. We can just manage dive plans. Sometimes I think we're a little too connected. Like Wi-Fi calling, it's handy, but I'm still a little bit skeezed out about it, personally. Yeah.
Yeah, no, I keep mine off. I turn it on when I need it, and I was like, and that's off.
Just a note for you, Giselle.
Yeah.
I got a great shot on Atalanta.
Yeah.
If you wanted to
highlight
that.
Yeah. Ooh, okay. I got that.
Yep. Zooming.
That's good.
Okay.
This is hard.
Yeah, I bet. Oh my gosh.
Oh no, bumping into the sponge.
It's okay.
Is it?
Oh.
Oh.
Hello,
No,
sponge.
it isn't.
No, it doesn't. It was fun for a while.
Very up close and personal.
Yeah. Oh.
All right, can we zoom out?
Out. Okay.
I mean, if we could get a toe hold up on top of the ledge, we might be able to slurp a bit. You know, from just up here.
After this one critter.
So. Yeah.
Yeah. Oh, we might be let the critter go and just get the sponge.
2-for-1 deal did sound nice though.
Sponge, right?
Yeah. I might have gotten eaten, chewed up.
Oh.
Because I had the suction on the whole time. Because I just didn't know when the moment was going to happen.
Right, yeah.
I don't either. Okay, can we zoom in again?
Okay, going in.
Get out of there. It's a bycatch. Yeah.
Ooh.
Oh, come on. Oh.
Oh. Yeah, looks like we got something there, maybe. Is it in the jar? I don't see anything in the jar.
I don't see anything in that jar.
If you want to just get sponge, we can go up top. I think this is too much.
Yeah, that angle is difficult.
Okay.
Can we zoom out on Atalanta?
Yep.
Yeah, you need to come. Yeah.
Yeah, that's full wide on Atalanta.
All right, I'm just gonna go plant up top. Is the camera? Can you rack it out? That'll help. So any of this is good, right?
Any of the low-B one.
So this one, correct?
Yeah. That's the one.
Yeah. Maybe there's another one of those things up here.
There might be.
Is that one right there?
No, it's just like a dead piece.
Just a dead piece here?
Maybe.
There's something pinkish.
Oh.
Can you tilt down a little bit and see the basket?
Oof. There we go.
Might be a shrimp. You want to see the basket?
Good for a zoom there.
I think I gotta move the arm out of the way.